Thank you so much. It's actually always humbling to be able to come and talk to you all. I am going to apologize in advance because I do have a second talk in one of the crystal ballrooms on molecular markers. And I unfortunately will not be attending the dinner tonight, but I will be here tomorrow. And I plan to hang out at the MEN2 registry table, or you can find me on the first floor somewhere. Many of you, I've talked to many of you already, so that you know that you just come up and talk to me, and I'm happy to answer any questions that you might have. I have a lot of information to present today, and what I'm going to charge you with, those people that are here, I'm told that they're recording this, they're going to put it on the website. If you find it useful, if you find it great information, you want to pass it along, spread the word. When they go up on the website, other people can access it. I've had my presentations posted on the Medi site before, so it's there for educational purposes. Some of you are going to go, what the hell is he talking about today? I'm sorry, I want to try and hit sort of a middle ground here, but I'm happy to go into details on certain things. I was really bad because every year people ask me to add more stuff, and so I'm up to, I think, 60 slides. So let's get into it. So my goals for today are really first to give you a basic introduction of tumor biology, because let's face it, cancer is cancer, and there is an underlying commonality to all cancers. Then what I want to do is talk about the role of gene mutation in medullary thyroid carcinoma. We know a lot about the mutations and their role in the development of this disease, and then shift into talking about how mutation data is used in clinical decision making. And if I end really early, we can have questions and discussion today. If I don't, then we can have them at the end of my second talk or tomorrow. So the first thing everybody should remember is that actually cancer is caused by genetic mutations. It's not just one mutation, it's actually a series of mutations. Having a single mutation is typically not enough to turn a normal cell into a cancer cell. You actually need to have multiple events that occur that finally convert that into a full-blown malignant cancer cell. And in fact, some time ago, back in 2000, a very famous cancer researcher, Robert Weinberg, proposed that all cancer cells have to acquire 6 different features in order to convert from being a normal cell into a cancer cell. These are those 6 different features. The first one up here is that they had to develop the ability to grow. I mean, we all know cancer cells grow, so we need to have a new ability to grow. We also have to develop an ability to ignore stop signals. Normal cells know when to grow to replace damaged tissue and when to stop because the damage is replaced. Cancer cells don't know when to stop, so they ignore that. Cancer cells also have to learn how to spread to different areas. That's the word that we hate hearing, metastasis. That is a trait that normal cells usually don't have, being able to spread. Here's the horrible thing. We all want to live forever, but we don't want our cancer cells to live forever, and that's a trait that they develop, is this ability to live forever. They need to generate a source of food, and that source is blood and oxygen. So they generate this source of food, and then they develop protection. They are no longer subject to the normal mechanisms that exist for cell death. So these are the 6 features that are required for a normal cell to become a cancer cell. Here's an example. We have a primary tumor here. One cell, so all of a sudden, grew into a bunch of cells. But the blood supply is far away. And in order for the cell to be able to grow into a larger and larger tumor, it needs to attract new blood vessels, a new source of food, in order for that tumor to grow. And in fact, there are therapies, anti-angiogenic therapies, that are based on blocking this process. And for those of you that know about the therapies for thyroid cancer, they in part block this process. So when we want to create a cancer cell, we don't do it all at once. As I said, it's an acquired of mutations, and they occur typically in a stepwise fashion. And there's no given order in which all of these 6 different targets are hit. And you can actually have multiple hits occurring that are not sufficient alone. Or you can have one gene that when it's mutated might actually affect 2 of these processes. So that becomes an important consideration, when you're thinking about the biology of a cancer cell, that it's a stepwise process, that you need to have all of these events occur in order to turn the normal cell into a cancer cell. Now, 3 years ago, we decided that, in fact, Dr. Weinberg went back and rethought his process, and he added 2 additional hallmarks or requirements of cancer cells. One of them is that they looked at energy differently. We all know that cells love glucose. That's why we have sugar in our veins to feed our cells and to create energy. Well, tumor cells actually have a different energy balance than normal cells. And the other thing that has been found is immune response. Everyone's been talking to me about immune response and immune therapy. And why is it that there's so much talk? Well, it's because cancer cells are learning how to evade the normal tumor response. Tumor cells actually are being found by the immune system in your body, and they're being eliminated. So normally, early on, before they acquire all of the mutations necessary, they're being recognized by the immune cells. An immune cell called a natural killer cell, or immune cells called T lymphocytes, recognize these cancer cells, and they will destroy them. But as the cancer cells develop more and more mutations, they become more resistant to being destroyed by the immune system, and they develop tricks to figure out how to not be recognized. There are what's called an immune checkpoint. You don't want to be killing every single cell in your body because you might kill good cells. That's how autoimmune diseases occur. And so you want to have checkpoints in place to only kill cancer cells. For those of you that really want to learn more about immune therapy, I would recommend going to this website here, which is called fightcancerwithimmunotherapy.com. I found it last night because everyone was asking me questions, and I give it my seal of approval, even though it's a drug company, to go there and learn more about immune therapy. So the bottom line is that a single mutation really doesn't cause cancer, and it's very, very complicated. So complicated that no one's going to walk out of here today understanding what caused their cancer or their loved one's cancer. So what are the mutations that initiate or drive the process of MTC? What do we know about them? And what are the additional mutations that cause this progression? I've said you need more than one mutation. What are they? What I can tell you with 100% certainty is that if you inherit an activating RET gene mutation, you are at very high risk to develop MTC. That's the only thing that I can tell you with 100% certainty. We know that if you have inherited or passed on a RET gene mutation that activates that gene, you are at high risk for MTC. RET is actually a receptor tyrosine kinase. Its role in this process of MTC was first discovered in 1993. Lois Mulligan and Bruce Ponder out of the UK, with the help of a variety of consortiums, found that mutations in RET are what cause this hereditary form of MTC. 1 year later, in 1994, it was discovered that a high percentage of sporadic MTC actually contain RET mutations. That linked RET as a cause of MTC. In the laboratory, we have human cells that we can grow and culture that are MTC cells. They have activated RET, and when we inactivate the RET in those cells, it actually causes the cells to grow slower. Doesn't kill them, but they grow slower under the microscope. You can see them all of a sudden arresting their growth. And we know now from the past 6 years of studies that in clinical trials, if you use drugs that were developed to inactivate RET, that MTC growth slows in a lot of people, what we call progression-free survival, PFS. And drugs like imatinib, mitasinib, sorafenib, which I heard when I walked into the room, which is also Nexavar, they slow cells that are being driven by RET. So what does RET do? Well, RET is actually a communicator. It receives signals from the cells that are surrounding it, and it receives those signals, and those signals tell the cell what it should do. What are the signals, and what is it telling the cells to do? Well, it's actually rather complex, and I've tried to make it simple here. RET exists in the membrane of the cell, so it is sticking outside of the cell on the outside, and a molecule will come along and it will bind to the RET. And this is a specific molecule. It's called a ligand. It is the thing that signals RET that it's time to do something. Once the molecule binds to the RET, the receptor then becomes activated. How does it become activated? We need two of them. They need to be close together, they need to bind, and this creates a process called phosphorylation. And this phosphorylation event activates a cascade inside the cell that sends a signal throughout the cell, including the nucleus, through a variety of complex things down here that ultimately lead to cell proliferation. What do these mutations do? These mutations remove the need for this signal. So think of it as when you have sugar, you've got a lot of sugar around, so you're very energetic and you know that you've got plenty of fuel to run around. But if somebody flipped that switch on and gave you all this energy without having sugar around to tell you that you could do it, you're going to run around crazy. And that's what these mutations do, is that they allow the receptor to have their signals stuck on. So they're always giving the signal to the cell, and they're always telling the cell it's time to grow. The MEN2A mutations actually act to turn the signal on by allowing two of these receptors to form a dimer, to bind together. The MEN2B signal, the 918 mutation that's quite common in sporadic cancer, actually activates the phosphorylation pathway, leaving the signal on. So, question, if I have a RET mutation in all of my cells, why is it that only my thyroid's affected? I mean, you have a germline mutation, that means every single cell in your body has that mutation, and that you can pass that mutation on to your children. I get asked from time to time, why is it that that's happening? Well, first of all, you are at risk for developing other types of cancers. For those of you that are affected by MEN2, you know that there are a high risk for developing parathyroid disease, for pheochromocytomas of the adrenal gland, and if you have MEN2B, for developing nerve tumors that exist throughout your GI tract. So it is not only your thyroid that is affected by having a germline mutation. So why is it? Well, it turns out that the RET gene is not expressed in every single cell type in your body. It's only expressed in certain cell types. So you need the gene to be expressed in those cell types in order for it to act and change the proliferation. So we can see from this, or if I take my glasses off, that it is expressed in the adrenal gland. It is expressed in the colon. It is expressed in the thyroid. But it's also expressed in the liver. It's also expressed in breast. It's also expressed in the testis. But we don't get tumors there. So we are fortunate that at least where we know the gene is expressed, it doesn't always cause cancer. Well, why is that? Well, remember I said that you need multiple mutations. RET isn't by itself sufficient to cause cancer formation. So the ability to introduce additional mutations is actually environment dependent and location dependent. For some reason, your thyroid is one of these areas that you have a higher risk for getting additional mutations. And perhaps because of that and the environment of the cell, it becomes the highest risk organ for developing cancer, whereas other tissues are at lower risk, but we still hear about them from time to time. Okay, so at least half of the room is going, but I didn't inherit a RET mutation and I got MTC anyways. Where did that come from? Well, some of you have heard about somatic mutations. Somatic mutations are mutations that occur only in that one tissue, in this case, only in the thyroid C cell. They cannot be passed on to your children. Important, remember that. If you're told that you have a mutation in your tumor and you've had your blood tested and it's not in your blood, you can't give it to your kids, okay? That's important. But how did you get that mutation? Well, mutations actually occur all of the time. They occur when DNA is copied and cells divide. They occur when you're exposed to mutagens, and they just build up over time. So most of the mutations we're getting mutated right now. Probably this radiation coming off of this tiny little computer is causing mutations. I have cells that are replicating in their mutations. But guess what? I mean, the human body is a wonderful machine. It recognizes that there are mutations and they shouldn't belong there, and there are machinery in place to get rid of them. So while you will develop millions of mutations over the life of your lifespan, you will probably, through estimates of science, only have about 50 to 100 new mutations that you possibly could pass on to your children. And most of those mutations may do nothing. Some of the mutations will be so bad that they won't be viable and produce children. The other thing to remember is if you have a mutation in a cancer-initiating gene, it actually may accelerate the rate that you get additional mutations and allow you to then hit all of those other hallmarks. One of the interesting studies that I talked about 2 years ago was somebody looked at the frequency of MEN2B mutations in male testes. These are from deceased men. They didn't take the testes from living men. So we're not asking for volunteers. But these are individuals that had no cancer. But if they looked for the MEN2B causing gene, they found that it existed at very high frequency. Anytime you see red or orange color, that is a section of a normal male testis that they found the 918 mutation. And it existed at 100 to 200 times higher frequency than statistics would predict. This is probably the main reason why so many patients with MEN2B are de novo. Neither one of their parents have that mutation. So, is the RET mutation involved directly in the induction of the cancer hallmarks? I can tell you that we really don't know a lot. I'll tell you what I know. It's not an active area of research. There's not a lot of money dedicated to MTC research, but I think that we've learned a lot over the several years, and we have some very wonderful scientists to thank for actually discovering that RET is a main driver, because before that, we had less knowledge and ways to treat it. So clearly RET is involved in sustaining proliferation, up at the top of the list here. We know that for sure. One of my colleagues at MD Anderson received a ThyCa grant. With that ThyCa grant, she had the question, does RET play a role in evading cell death? And she worked very hard for 2 years on that and has actually discovered that RET does play a role in preventing cancer cells from dying, and I'm hopeful that that paper will eventually be published. But what it says is that when you inhibit RET, you actually help activate mechanisms of cell death, and that RET functions not only in causing cells to proliferate, but RET also functions in preventing them from dying like normal aberrant cells would die. Dr. Gagel, who spoke yesterday, has some work that I did along with him that's currently unpublished that suggests that RET actually may play a role in genomic instability, the progression of additional mutations. In fact, what we did when we compared DNA isolated from sporadic tumors and hereditary tumors, looking at those wonderful tools that correct mistakes. Whenever you get a mutation, we have a machinery of almost 200 genes that exist in your body that function to correct mutations and mistakes. It turns out that if you look at those 200 genes, a lot of them are deleted in sporadic cancer, meaning that these precancer cells are less able to correct mutations when they occur, and that allows us to move on and finish out all of those hallmarks and, in fact, become a malignant cancer cell. There's some ongoing work that has been funded. One of them was funded to Dr. Grubbs, who will be speaking tomorrow, looking at the role of RET in evading growth suppression. And that's, normally cells have mechanisms to tell them to just stop growing. And Dr. Dadu this year, Ramona Dadu, some of you have probably heard her name through the CASAD study. She is a new faculty member at MD Anderson that received a ThyCa grant this year to start studying immunotherapy. No one has worked on immunotherapy in MTC. We don't know if it will work. She's doing studies to ask the question, will it work? And I have some work that has been ongoing looking at whether or not RET plays a role in immortality. So I can show you this. I showed this 2 years ago. This is an assay that we do in the laboratory. It uses human medullary thyroid cancer cells. And what we do is we grow them in a culture dish. And I think maybe you can see these little blobs up here. This is an assay that measures immortality. When these cells exist in blobs and you can count these blobs, that means the cells are immortal. We know these cells are immortal because they're cancer cells. If we remove RET, guess what? Less blobs. That means that RET is playing a role in the immortality. Now, another thing that plays a role in immortality is these growth factors, FGF and EGF. And so if you do the triple whammy, which is what's shown here, you actually can prevent and take away the ability of these cells to be immortal. So because RET appears to be involved in so many hallmarks, it may be that if you have a RET mutation or are born with a RET mutation, you need fewer additional mutations in order to get cancer. Because we can show, or at least infer, that RET is involved in at least 5 of these hallmarks. And so it's unfortunately a bad gene. I can't say it more easily. All right, now, possibly the majority of you here, but I wasn't told that I had a RET gene mutation. What else causes MTC? I spent a lot of time talking about RET. We all know about RET. You're all wondering about RET, but what about other genetic drivers? Well, the way we scientists, and you can do this as well, find out about other mutations that exist in cancer and other mutations that might be involved in driving cancer is this tool that was generated out of England, the Sanger Center, called COSMIC. It's called Catalog of Somatic Mutations in Cancer. And what they do is they have a website that catalogs all of the reported and unreported mutations that people have found sequencing tumors. You can actually go in here and specifically look at medullary thyroid carcinoma and ask, what mutations have been found in tumors from medullary thyroid carcinoma? Well, here's the list. RET is at the top. Of 1,549 tumors that have been analyzed for mutations, 625 had RET mutations, 40%. So 40% of somatic, not hereditary, MTC has a mutation in the RET gene. But guess what? There are other mutations found. If you go down and look at the next 3 genes, they all have the word RAS in it, R-A-S. There's HRAS, KRAS, NRAS. There have been fewer tumors looked for this. It's 550, 560 tumors. But what you find is 10% of it, 9.3% HRAS, 3% KRAS, not quite 1% NRAS. BRAF, for those of you that have been attending the papillary, you heard about BRAF. BRAF have been found in about 10% of tumors, but there's a caveat here that a lot of us that do research wonder if this is a true finding or not. And then from work that came out of Johns Hopkins and that was supported by the MEN2 consortium, the only study to date that has done full exon sequencing of MTC tumors have found a couple other genes that might play a role. Barry Nelkin and his group at Johns Hopkins found VHL mutations and MDCA1 mutations are associated with some frequency. And I'm not going to talk about those today because we need to really get the numbers up to see whether they're important or not. So RAS is now number 2 on the hit list for those people out there that are going, I don't have a RET mutation, what might I have? RAS might be the mutation. We've done a study at MD Anderson in which we've now up to 150 patients looked at RAS mutations, and we're actually finding the same thing. About 13% of the patients seen at MD Anderson have RAS mutations. About 40% of the patients have RET mutations. So RAS is now the second known driver for MTC. We know RET's a driver, we know RAS is a driver. Does this make sense that RAS is a driver? Well, actually, when RET is turned on, its first job is to turn on RAS. Sorry I don't have a laser pointer, and the mouse on this is not very good. But so you see that RET gets activated, it turns on RAS. RAS gets activated, it turns on BRAF, and then it all goes all the way down. And so because RAS is downstream, what that suggests is that we're hitting the same pathway. For those of you that have followed the other cancer, what is that called? PTC and FTC. We know that they also have RAS and BRAF involved. So it's the same pathway. RAS is actually involved in a small group of GTPases. What's interesting, and I didn't even know this, is that RAS was first reported even before RET as a possible driver for MTC. Back in 1989, it was found that RAS was elevated in some tumors. The more recent studies that went back and looked at RAS found that most tumors that are RET negative were in fact RAS positive. In this one study published in 2011, they found that 68% of non-RET tumors were in fact RAS positive. That number has actually been debated. We think that it's a lot lower. I just showed you the 13%. And so we don't know. And what we also don't know is what is the role of RAS mutations in prognosis and targeted therapy. Because it's so new, we're trying to figure out what these RAS mutations mean. One of the things I clearly want to point out is that these RAS mutations are the same mutations that are found in other cancers. RAS is one of the most common mutations found in the cancer world. In fact, 16% of every single type of cancer has been shown to have a RAS mutation. So it's a very common mutation. It's very well known to cause cancer and very well associated with cancer. Despite this, we don't have targeted therapies for RAS. It's been a huge, huge problem in the oncology group, is finding something that can target RAS. So let me just give you this very, very simplified view. RET, 41%, RAS, 13%, other mutations based from the Johns Hopkins group, probably about 10%. That leaves 36% of people with MTC. We just don't know. And I'm sorry, I can't give you an answer to that, and we're still looking. We're trying to figure out what is in that group. We've got a few nibbles here and there, and hopefully we'll figure that out. But it's not all bad news. I mean, there is research going on looking to address this. So what can we do with this mutation data? Well, the very first thing that I want to tell you is that utilize this information with great caution, okay? The current guidelines that we tell all of the physicians, and they're supposed to be following, although some don't follow these guidelines, do not address specifically whether or not you should treat a patient based on whether they have a mutation, somatic mutation, yes or no. The only thing we know is if you have a germline mutation, there are clear recommendations. Somatic mutation, we don't know. Just because you have one of these mutations does not mean that you should immediately start taking one of the drugs that are developed to target this mutation. These drugs are reserved for slowing the growth of tumor when the tumor is growing. If the tumor is not growing, you're not going to have any benefit from taking these drugs. And again, honestly, anyone that gets diagnosed with MTC, I strongly, 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 strongly recommend that if you know anybody, tell them to get their germline tested, because the best way to cure MTC is to cut your thyroid out before it develops into cancer, and the only way we can do that is if we know you're at risk and you've inherited the mutation. So, mutations and treatments. There are currently 2 approved drugs that target RET. I've listed them here as vendetinib and cabozantinib. They're also known as Calpressa and Cometrix. And what they do is that they target RET, this receptor up here with at the top, but they actually also target other receptors. The number in the box is the efficiency at which those drugs act to inhibit those other receptors. The VEGF receptors, VEGF receptors, those receptors play a critical role in blood vessel development. So we know that if you're taking either one of these 2 drugs, you're actually also inhibiting angiogenesis, one of those important factors required for cells to grow. The 2 drugs act differently with response to EGFR and MET. EGFR is thought to play a clear role in proliferation. MET is actually thought to play a role in metastasis. So we don't know if inhibiting MET plays a role in the metastasis of MTC or not. The bottom line is neither one of these drugs target RAS. RAS is below there, you see. So if you're taking these drugs and you have a RAS mutation, you're really not targeting it. So, but are they effective? So if we look at the clinical trial data that was published that got these drugs FDA approved, what you just heard me say is that if you don't have a RET mutation, then you have a high chance of having a RAS mutation. So if we look at the patients that did not have a RET mutation in the study, that's around that's highlighted in the box, we really only had 2 patients on that clinical trial that received vandetanib that might have had a RAS mutation. And if you are on the way you read this diagram here is, if you're on the left side that says drug better, that means that you're getting better on the drug. And you can see that when you don't have a RET mutation, it's right down the middle. Some people are doing better on the placebo and some people are doing better on the drug. So from this study, we don't have enough information to know if vandetanib helps patients that have RAS mutations. We also don't know if you're on cabozantinib or Cometrix if you do better, because you can see, and here the group is a little bit bigger, and at least there is some positive effect, but there's also patients that don't do well. Well, here is where I'm actually going to throw out a positive plug. The Exelixis decided, when they found out that RAS mutations occur, to go back to those samples and ask, in the RET negative samples, are there in fact RAS mutations? And it turned out that they analyzed 85 samples, and in those 85 samples they found 16 patients that had RAS mutations, and then they went back to that data set and asked the question, did those patients respond to drug? And this is progression-free survival, and what you should note here is the pink line is 13 patients that were on drug. The blue line is 3 patients that are on placebo. All 16 of these patients had RAS mutations, and what you can see is the drug actually had the same effect on progression-free survival as patients that had RET mutations. So the whole group, all 330 patients, had a progression-free survival of 45 weeks. If you had a RAS mutation, you had a progression-free survival of 47 weeks. So what this says is that this drug works even if you don't have a RET mutation, so that it's not a bad thing to be put on this drug. And what it also says is maybe targeting, looking up your mutations and knowing that information is not the best thing at this point in time. We just don't know. We're still learning. This is data that is not published yet. So is there any support for actually targeting RAS? We can't target RAS itself. We know that people have RAS mutations, but we do have the ability to target the downstream pathways here. There are drugs that can come in and act below RAS, and perhaps they could be used in those patients that we know have RAS mutations. In cell culture experiments, what we find is, in fact, that if you use a drug that hits one side of the pathway or the other side of the pathway, the cells actually die. And if you mix the two drugs together, the cells die even better. That's what these lines going down mean. And if you put these tumors in mice, I've already spent most of my time saying how I don't like mice studies, but if you put these tumors in mice and you grow them, if you give them no drug, you see how the untreated bar stops at 10 days? That's because they had to sacrifice the mouse because the tumor was too big. The animal care facility said, you're being inhumane, we need to put the mouse down. But if you give them the two drugs, the mouse is allowed to go out, out to 20 days here, and the tumor is growing much slower. So we know that targeting the RAS pathway does help. Unfortunately, these drugs are not available clinically. These are all drugs that exist only for testing in cell culture. There's a couple drugs that do exist clinically that target this. One is sorafenib. It targets RET and BRAF. So BRAF is downstream of RAS. Another drug is selumetinib, which targets downstream as well. What I want to do is I want to focus your attention on the red circles. So where you see the red circles that are high up here, that's when you add just one of those drugs. One of them is sorafenib, one of them is selumetinib. When you add, and the higher up on this, that means the cells are growing with and replicating fast. When you add both the drugs, boom, less growth. For those of you that were lucky enough last year to hear Ross Kagan talk about his drug cocktail work, his approach has been similar to this, targeting multiple arms, mixing the drugs together, because then you can use lower concentrations and have a more effective response in preventing cell growth. These drugs are FDA approved, but there aren't clinical trials to test them. We actually just got turned down by NIH to start a clinical trial on this. Some caveats, though, is that both of these cell lines that we use have RET mutations. They're not RAS-driven tumors, they're RET-driven tumors. But remember, RET's upstream of RAS, so perhaps they are okay to use. But it really will probably take clinical trials to assess whether or not these drugs are really good at targeting a RAS. So, actually, I think I did not so bad. I'm going to give you some factoids to take home. So first of all, we now know RET and RAS mutations drive MTC formation. That's clear. We know that treatment for advanced MTC exists because when we found these mutations, it led to development of drugs to try and target these mutations. And that was the first step of being able to provide new therapies for patients where there weren't therapies before. We currently have these therapies, vandetanib and cabozantinib, and I will point out that while they target RET, they also target angiogenesis, they also target other pathways, and this is why probably they are effective. And it's probably clear that angiogenesis is very important because in tumors that have RAS mutations driving the cancer, these drugs work. So those tumors are not being driven by RET. RET is not being inhibited in those tumors. The only thing that's being inhibited is the angiogenesis. We do think that trials incorporating BRAF and PI3K and MEK inhibitors may be a better way to facilitate treatment of patients that specifically have this new class of RAS drivers. But again, what's important to remember is we don't have clinical studies or data that would suggest that we really will have a beneficial outcome by targeting these mutations. These are all just laboratory studies done in mice and cell culture. Logically, it makes sense to do it. They're very encouraging, but until studies are actually done in patients and they show that these treatments work better than other treatments, we really don't know. And again, I really need to say that if your tumor's not growing, don't even think about this right now, because there's a lot more that we don't know about these drugs than we do know about them. And probably the most important thing is unfortunately we really don't know what's causing MTC in nearly half of these patients. We still haven't identified what the underlying driving cause is. So that's an area that we need to continue to invest research effort in. So I want to acknowledge all of these wonderful people that have been involved. Dr. Grubbs will be here tomorrow. Dr. Gagel I have worked with for 30 years. And there has been, now there is funding through the NIH, I think, and a lot of that is due to support from advocacy groups. I am part of a NIH SPORE grant that is several $1 million a year for the next 4 years. This is our first year that will look at mechanisms of drug resistance and develop new ways for following MTC proliferation. And so that's an important step. And the American Cancer Society has been a godsend for developing new research strategies for treating MTC through the MEN2 consortium that has directed research specifically for MTC. So I'm going to stop here. I'm sorry. We
I
have,
think we have about 20 minutes for questions.
really?
Well,
3:30?
We go to 3:55?
Did I really talk that
You're
fast?
a little bit longer.
I am. Well, then that's great.
That's great. So let's open it up for questions.
How did I talk in 45
I
minutes?
don't know how you did it.
And how long was I supposed to talk? Oh, I got over an hour.
Oh,
Okay.
yeah, you're gaining time each conference.
I didn't take a breath. Okay, in the back. You've got a choice. You can speak loudly and I'll try and repeat your question.
I was asking you to just go back and put the slide up your 2 slides ago.
This slide?
Yeah.
Okay.
Thanks.
Oh, you want to write down what's there?
I do.
Okay. Okay. Yes.
I have a question. Is there anything to keep oncologists who are not in the Center of Excellence from prescribing the drugs that are out that, to their patients? I started out someplace other than MD Anderson, and I can remember having conversations with my oncologist where I would ask him a question, he would just laugh. He didn't no idea. But I think if these drugs had been on the market, he would have put me on one.
And that's why you heard me say, so the question is, how can we stop or slow physicians from overprescribing these drugs? And it's a very important question. I will, first of all, I need to repeat and reiterate, I do not see patients. I am not a physician. A lot of what I'm going to say is my own personal opinion from being around physicians and interacting with everyone in this room and having come to ThyCa. The first thing that I want to say is, right now, to be able to prescribe either one of these 2 drugs, you actually have to pass a test from the drug companies. It's a black box examination that deals with one of the side effects of these drugs, which is arrhythmias of the heart, the long QT. That's the only obstacle in the way of preventing physicians from prescribing these drugs. The other obstacle is you guys out there right now and people like me that will say, please tell people that these are drugs for people that have progressing disease and have no other choice. We all know somebody that was diagnosed with, well, in this room, we all know somebody that was diagnosed with MTC, and it's just sat in their body for 10 plus years doing nothing. You don't want to be giving people these drugs that the MTC is doing nothing. That first of all, they're really expensive. We don't know what the impact of giving these drugs on patients is long term. What we can tell you is short term, if you have progressing disease, the disease is slowed. And who wouldn't want their disease slowed if it's progressing? But patients need education as well. They need to recognize that if they have stable disease, they shouldn't be asking to be put on these drugs. We have, I'm fortunate that at MD Anderson, the physicians, they probably wait a little longer than, a lot of people before they'll put people on these drugs because, we only have 4 or 5 years experience with these. We have 50 years experience of watching people with living day to day with metastatic disease. And, we have people that have MTC with metastatic disease and die of other natural causes. You need to remember that. And so I personally can't do anything to stop people from overprescribing this drug. The drug companies obviously want everybody to take this drug because they invested a lot of money making it. That's why it costs so much money. So if you guys have suggestions, I'm more than happy to. What's probably going to happen is you're going to go, go to ThyCa website. Dr. Cote said no one should take this drug. And then the drug companies are going to look me up and...
I think that Dr. Sherman earlier today made a really good point, especially for newbies, and that is that you think of metastatic disease as being aggressive and therefore needing treatment. But his point this morning that he made was that medullary, along with other cancers, tends to metastasize very early on. Way longer. It takes a long time before it's detected. So the fact that you have metastases doesn't mean that you have an aggressive form of medullary, and doesn't put you into that automatically into that classification that needs treatment with these diseases just because you have a metastasis.
Correct. So let me reiterate. The comment was made that remember that MTC is something that metastasizes early. Remember that be thankful that there is actually a way in which it's found. Because it's in your throat and you feel a lump there, you often go early and get it looked at. Think of these poor people with pancreatic cancer. They can't feel a lump down here, and by the time they have symptoms, that disease is so widespread that there's nothing that can be done about it. So it is a slow-growing cancer.
But it's early spreading.
But it's also an early spreading cancer. Where I would say that it would be wonderful to have research and to figure out, the big question is, what happens that makes that disease that's sitting in the body all over the place, all of a sudden go, guess what? I want more real estate, and I'm going to start growing and spreading all over the place. We have no idea. I mean, there's honestly, and that's the event that's life-changing for a lot of you, because that's when you're seeking out these drug therapies to try and figure out, can you stop this thing that just happened? We don't know why it happened, but it's happened, and we're trying to slow it. We're trying to figure out what's going on. There's a second trigger that has somehow happened and now told the cells that it's time to take up more real estate, and that's what we just don't know. It could be that as we get older, our immune system becomes less and less effective, and that the immune system that was keeping some of the cancer cells in check is no longer able to do it. You know, hopefully studies that are looking at the immune checkpoint pathways will help us a lot, will allow us to figure out if medullary is one of these cancers that will benefit from boosting the immune system at these times. We just don't know. Yes?
You mentioned that you applied for an trial and it got turned down. Why did it get turned down, and you plan to follow up on
Okay.
that?
So one of the question is about new trials for MTC. Part of the NIH SPORE that was funded last year is one of the goals of the SPORE is to initiate a new trial that uses two drugs to try and hit the pathways simultaneously. So that trial is too expensive to run based on the funding that we receive from NIH alone, so we have to go in to the other agencies to get money through NIH to pay for the drugs. And it costs a lot of money to do a trial, and they want to make sure that they're not going to be wasting taxpayer money. So we applied to get the drugs, and we were turned down. Why were we turned down? Because the trial design was based on these studies from Dr. Matt Ringel at Ohio State, and these studies don't include animal models. So what they told us is, if you go back and repeat these studies in animals and show that in animal models you have this same effect, then we will let you move forward with the trial in humans. So that's currently the plan, is to actually do the additional studies that NIH has asked for, and then reapply for the drugs to do the trial. And so hopefully next year we'll be able to start that trial. So I was probably being a bit strongly worded when I said we were turned down. Yes, the back.
Yeah, on one of your previous slides, you were talking about the vendetta and how it interacts with, keep going back, how it interacts with the angiogenesis. And
Correct.
also, not killing the cell, not killing the cancer. So can we say from that not to really expect the cells, or not to expect the tumor to shrink very much?
So that has actually been a long-standing debate. I'm sorry. So the question is, does vendetinib kill and shrink the tumor? And from a purely scientific point of view, if you just, from a basic scientist, what vendetinib does is, in part, it inhibits RET. We know that if we inhibit RET, we inhibit growth of the tumor. We would not stop the tumor from growing more, but we wouldn't induce the tumor from shrinking. What we're now learning a little bit more through the ThyCa funded research from Dr. Yarman is that by inhibiting RET, we actually also increase the sensitivity of the cell to dying when it's exposed to agents that cause cells to die. So that would be one mechanism by which the tumor could shrink. Probably the biggest method through which the tumor shrinks is that vendetinib actually will cut off the blood supply to the tumor. And if there's no food, you're going to die.
Yeah. Okay, and I'm talking about that. Does it inhibit new blood supply, or does it leave the established blood supply there?
It really inhibits the new blood supply from growing. And there are constant signals that go back and forth between the tumor cells and the blood cells for when things become reestablished. One of the problems with tumor growth is that it kind of grows out in a ball, and you can't really get blood supply into the middle of the tumor. And so that part of the tumor has no oxygen. And so that's where the change in metabolism changes, where the tumor learns to grow in the absence of oxygen, but it also induces some cell death. The imaging technology, when you get your CAT scans, your MRIs, they don't know how to deal with the differences that are associated with blood supply and with lack of oxygen to the tumor. So sometimes the tumor will stay the same size, but they'll have different density when you're looking at it through the MRI. And those different changes, you might see a tumor that looks really gray, and then after you put it on drug, it starts getting black in the middle, or it'll change on the MRI. And that's sort of an indication that while the tumor hasn't shrunk or changed in size, it's actually the biology of the tumor has changed. And again, I'm not a radiologist, so I don't interpret those things. So you have to remember that some of what I could be saying is completely wrong. But I like to offer hope. So, Sure. Okay, another question, sorry.
My question's a little more elementary, but regarding men too, once the children have been tested for the mutation and say they're all tested negative, is that the end of the mutation for that bloodline? It's done, or are there other tests required?
Okay, so the question is, if you're a parent and an MEN2 diagnosed person, you've been found to have a RET germline mutation, and you want to know what is the risk that my children will have suffer the same fate that I have done? And you go and you do the RET testing, and you find out that they don't have the RET mutation. Are they still at risk? And so the short answer is they're done. The long answer is that they now have the same risk for developing cancer that everybody in the population has. The great thing about the discovery that RET was responsible for hereditary MTC is it gave us a way to cure cancer in individuals at a young age. One member of your family has to take the bullet. They're the ones that have to be discovered as having the tumor. Once they're found as they've had the tumor, there should be a massive effort to find out, is that a germline tumor? Do I have a RET mutation? Can we find the RET mutation? Once you find that you have a germline RET mutation, through a blood test, not through a tumor test, if they can identify a RET mutation, that becomes the cause of your tumor. Once they have that signal, it becomes possible to then say, was that signal passed along to any of your family members? If that signal was passed along to your family member, they have a risk of developing cancer. If you remove the thyroid early enough, they're cured. Dr. Gagel has patients that have had their thyroid removed based on calcitonin levels that have been disease-free for more than 40 years because of the ability to go in and act early. If you have not passed that gene along, they're clean. They don't have to go every year. They don't have to have calcitonin tests. They're done. Now, for those of you that are thinking about having children, I'm not one of those people, there is in vitro fertilization technology to guarantee that if you want to have children, that they will not inherit the mutation that caused your cancer. It's expensive. It's controversial because certain people feel like you're doing something that's not natural. But there is a technology out there to guarantee that you will eliminate cancer from your family forever, that type of cancer.
So can I just also say that it's the medullary part, by having the thyroidectomy.
Correct.
Be eliminating the risk of medullary.
Correct.
Not the other components of the.
Yes, but again, if you have a mutation, your kids don't have that mutation, you eliminate medullary, theo, everything, completely. For the patients that had their thyroids out before the age of 10 that are cured of medullary, they still have to be monitored for pheochromocytoma. Sorry.
We're running close, but I do have a question that came up on a group regarding the genetic testing. And it had to do with the confusion about saliva versus blood testing for genetic.
Okay. So the question relates to, if I'm going to have genetic testing to determine if I have a hereditary mutation, what should I have my genetic testing done on? Most people will go, well, why aren't you testing my tumor? Well, you can have your tumor tested, and they can find mutations in your tumor, but if they test your tumor, they don't know if that same mutation exists throughout every cell in your body. So they have to actually pick a cell that's not a tumor cell type to do that test. Most of the time, they will draw a tube of blood and isolate from the white blood cells in you the DNA, and because those cells are representative of other cells in your body. If any of you have ever watched a CSI program, they come at you with this long cotton swab and say, open your mouth, and they scrape it on your cheek. What are they doing there? They're actually scraping cells off of your cheek. We call it buccal cells in a buccal swab. Those are normal cells. Those are cells like your blood cells. If they isolate DNA from that and find a mutation in that, that says that you've got a germline mutation. Now, kids and individuals that don't exist near a genetic testing center, we often just send them out a little kit with a cup and say, spit in this. Call it a spit kit. Kids love it because they don't have to get a needle in the arm. And they have contests to see how full they can get the cup. In your saliva, there are the cells that exist on your cheek walls are dying, and being replaced constantly. Anyone that's ever bit their tongue knows that the next day, geez, the bite's gone. It's because the cells are actually dividing rapidly and the dead cells are being replaced. But the DNA actually is still floating around your mouth. We're all eating DNA. Ugh.
So that doesn't connect to this blood test?
It is.
Okay, then that answers that part perfect.
It,
You are
what?
close to the end here.
Okay, I'm close to
For
the,
your benefit, since you have to.
I have to get over there at 4:05.